In this video tutorial, I am going to discuss about the classification of proteins. Proteins are mainly classified in two different ways. The first one is the based on function of proteins and second is the based on chemical nature of proteins. So let's first look at the classification based on function of proteins. So here proteins are classified according to their functions, and so 8 different groups are available. The first is structural protein, second is the catalytic proteins, third is the transport proteins, fourth is hormonal proteins, fifth contractile protein, sixth storage proteins, seventh genetic proteins, and eighth is defense proteins. So let's first look at the structural proteins. Now structural proteins are those proteins which can help in the maintenance of structure of various tissue. So here we have two examples. The first one is the keratin, which is important in the structure of hair and nails. Second one is collagen, which is important in the structure of bone. The second subgroup is catalytic protein. Now catalysis means accelerating the rate of reaction. So those proteins which can help in accelerating the rate of other chemical reactions, they are classified as a catalytic proteins. And we all know that enzymes are the protein which can help in the catalysis, right? So enzymes are also known as catalytic proteins, and examples of enzymes are hexokinase, amylase, etc. Actually in our body there are thousands of different enzymes, and any enzyme can be named under these catalytic proteins. The third one is the transport proteins. These proteins helps in the transport of various substances in our body. For example, hemoglobin. It can transport oxygen and carbon dioxide, right? The second example is albumin, and probably albumin is the single most important protein which can transport so many different substances. So here albumin can transport bilirubin, calcium, free fatty acid and many different drugs. So albumin is also classified under transport proteins. The fourth category is the hormonal proteins. Now some proteins are there in our body which can act like a hormone. For example, insulin. Insulin is a hormone which can bring down blood glucose level, right? So insulin, it functions like a hormone, but if you examine it chemically, it is protein. The second example is growth hormone. Growth hormone, it brings about the growth of various tissue in our body, right? So it functions like a hormone, but chemically it is a protein. So insulin and growth hormone both are classified as a hormonal proteins. The fifth one is the contractile protein. Those proteins which can bring about contraction and relaxation of muscle, they are classified under this heading. So examples are actin and myosin. The sixth one is the storage proteins. Now this protein helps in the storage in our body. For example, O-albumin. O-albumin is the storage form in the egg. The second example is glutenin. Glutenin is the storage form of protein in case of wheat. The seventh subclass is the genetic proteins. Now there are in our body, there are certain proteins which can help in the maintenance as well as regulation of genetic material. Those proteins are classified as a genetic proteins. For example, nucleoprotein. And the last subclass is the defense protein. In our body there are certain proteins which can help in the defense of our body system. For example, when bacteria or virus invades our body, at that time our body produces certain protein, and these proteins are called as a defense protein. For example, immunoglobulins. We all know that immunoglobulins, or you can say antibody, they help in killing of virus and bacteria. If we consider snakes, in case of snakes, how snakes defend themselves? Snakes defend themselves by its venom, right? So snake venom is also classified as a defense protein. So with this classification based on function of protein is complete. Now next we are going to look at the second system of classification, that is classification based on chemical nature of proteins. So now under this system of classification, proteins are divided into three groups. First one is the simple proteins, second one is the conjugated proteins, and third one is the derived proteins. Let's first look at the simple proteins. Now as the name suggests, these simple proteins are really simple. they are simply protein. There is no anything else other than proteins. So we can say that they are composed of only amino acid residues, right? And these simple proteins are further subclassified based on shape into two more groups. The first one is globular protein and second one is the fibrous protein. These globular proteins are the globules or oval shaped protein. That means their length and their width are almost identical, right? So they attain or they get a globe or oval shape, right? The second subgroup over here is the fibrous protein. As the name suggests, they are fiber-like. That means their width is very small as compared to their length. So they look like a fiber. So that's why they are called as the fibrous proteins. Now these fibrous proteins, they are also known as scleroproteins or albuminoids. Now remember over here is that albuminoids, it is different than the albumin. Don't ever confuse between albumin and albuminoid. Now the second category is the conjugated proteins. In case of simple proteins, we had seen that simple proteins are composed of only amino acid residues, right? Whereas in the conjugated proteins, amino acids are there. But in addition to these amino acid, there are certain non-protein moiety also there, which is known as prosthetic group or conjugating group. So we can write that in addition to amino acids, this protein contains non-protein moiety, known as prosthetic group or conjugating group. Now, if I want to show this in the simplified manner, I can write simple proteins plus non-protein group that makes conjugated protein. Now, here we have to understand the difference between these simple proteins and this simple protein. Remember, this simple protein, it has independent existence. That means independently these simple proteins are functional. But in this case, where simple proteins, it is occurring as a part of conjugated protein, they don't have separate identity or they don't have independent existence. Why? Because these simple proteins, they are not independently functional. They become functional only when its non-protein group is attached with the simple protein. And so this conjugated protein is a functional one, whereas its independent component, that is simple proteins, it is not functional. So to differentiate these two, this simple proteins has given another name, that is apoprotein, right? And its non-protein group, as I told you earlier, it is also known as prosthetic group or conjugating group. And when this apoprotein and prosthetic group or conjugating group combines, they make up conjugated protein, and this complete unit, it is also known as holoprotein. Now the third group is the derived proteins. Now derived protein, as this word derived, they are derived from where? They are derived from either simple proteins or conjugated proteins. Now the second question is that how they are derived? So they are derived either by denaturation or degradation. So if I want to write the definition of derived proteins, I can say that these are denatured or degraded products of simple and conjugated proteins, right? Now here we have to differentiate between these two words, denatured and degraded. Denatured means all the weak interactions of the proteins are broken down. Remember, all the weak interactions that include hydrogen bond, van der Waals forces or hydrophobic forces, right? But here strong bonds are not broken down. That means peptide bonds are not broken down in case of denatured one. Whereas degraded means hydrolysis of peptide bond is there. In other words, you can say that in case of denaturation, there is no any shortening of polypeptide chain, whereas in case of degraded or degradation, there is a shortening of polypeptide chain. So these two words has totally different meanings. So based on whether it is denatured or degraded derived product, they are further subclassified into two groups. The one is the primary derived protein, which is of the denatured variety, and second one is the secondary derived group, which is of the degraded variety, right? So I hope that by this you can easily differentiate that what is the meaning of simple protein, what is the meaning of conjugated protein, and what is the meaning of derived proteins, right? So next we are going to discuss about the various examples of all these three categories, right? So let's first look at the simple proteins, examples of simple proteins. And from the previous discussion, you know that simple proteins are further classified into globular protein and fibrous protein. So, we will first look at the examples of globular protein. So, we have 6 different examples under globular protein. The first one is albumins, second one is globulins, third one is protamines, fourth one is histones, fifth one is prolamines, and sixth one is glutellins. Let's first discuss about albumins. Albumins are soluble in water, and one characteristic feature of albumins it is that they are deficient in glycine and they are acidic in nature, which is evident by its pI value, which is pI is equal to 4.7. Example of albumins, that in case of animals, O albumin is there, which is found in eggs. Second one is lactalbumin, which is present in milk. If we talk about the plant sources of albumins, we have legumelin, which is found in legumes, and leucocene, which is found in cereals. If we discuss about globulins, globulins are soluble in dilute salt solution. Remember, albumins, they are soluble in water, whereas globulins, they are soluble in dilute salt solution. Examples of globulins: hemopexin, transferrin, then ovoglobulin, which is present in lactoglobulin, which is present in milk, and legumin, which is present in legumes. Right? The third example is the protamines. Protamines, they are soluble in water and dilute acid. The important characteristic feature of these protamines is that they are rich in arginine and lysine. Remember, albumin is deficient in glycine, protamines, they are rich in arginine and lysine. We all know that arginine and lysine both are basic amino acid, and the protein which is rich in basic amino acid, so these proteins, they are also a basic proteins, and which is again evident by their pI value, which is 7.4. So, whenever basic proteins is available, they have a tendency to bind with acidic substance. So, that's why these basic proteins combines with nucleic acid to form a nucleoprotein. For example, salmine, sardinin, and cyprinine. These three are the examples of protamine, and these three are found in fish, sperm, and testis. Then fourth one is the histones. Histones are very much similar to the protamines, but there are certain important difference. Histones are soluble in water and dilute acid, which is similar to protamines. Histones are rich in arginine and histidine. See, protamines were rich in arginine and lysine, whereas histones are rich in arginine and histidine. Okay. Now again, arginine and histidine both are basic amino acid. So ultimately, this protein is a basic proteins, and as we had discussed earlier, basic proteins has a tendency to bind with the acidic group. So these histones, they can combine with the nucleic acid and especially DNA. Now if we look at the example of histones, we have two examples, that is, the first one is nucleohistone and second one is the chromosomal nucleoprotein. Now let's discuss about prolamines. Prolamines, they have characteristic that they are soluble in 50 to 80% alcohol, and they are rich in proline, and actually that's why they are known as prolamine. See, from the proline, the word prolamine is derived. Why? Because they are rich in proline. Now their source is plant only. Okay. So we have gliadin, which is present in wheat, and hordein, which is present in barley. Right. See, wheat, this gliadin is the main member of this prolamines group, and that's why sometimes prolamines, they are also known as gliadins. Right. And the last subgroup is the glutellins. These glutellins, they are soluble in dilute acid, and they are rich in glutamic acid. And here also, this glutellins, this word is derived from this glutamic acid. Why? Because they are rich in glutamic acid, so glutellins word is used. Here again, this is also plant only source. So examples are orizenin, which is found in rice, and glutellin, which is found in wheat. So these are all the examples or all the subgroups of globular protein. Here I want to stress on one important aspect that you don't have to forget, right? See, the group name is glutelins and its one of the member is glutelin. Here also group name is gliadins and its important member is gliadin, right? Similarly, in case of these albumins, we had, as an example, we had not discussed about human serum albumin. Simply human serum albumin, we call them as albumin only. Okay. So what happens? These are the family names. These are the protein family names, and one of the most important family member has the same name as a family name. So albumins has a albumin, gliadins has a gliadin as a member, glutelins has a glutelin as a member. So we have to differentiate between the name of protein family and name of its important member. Both can be same, but both have a different meaning. In what context that particular word is used, that is important, right? Now let's look at the simple proteins, fibrous protein variety, right? Now there are certain important characteristics about the fibrous proteins. What are these? So fibrous proteins in generally they have very low solubility. We had seen that in case of globular proteins, they are either soluble in water or soluble in dilute salt solution or soluble in alcohol or soluble in dilute acid, like that. But in case of fibrous protein, it is not so. They are generally, they have very low solubility. So we never talk about the solubility of the fibrous protein. But this very low solubility, it has very important advantage. What is that advantage? Because of very low solubility, they have a great stability, right? As they will not get dissolved, right? So when they have a great stability, then it becomes an ideal tissue for the supporting structure of tissue. So we can write, so because of this, they forms a supporting structure of tissue. Now let's look at the examples or families which is classified under fibrous protein. The first one is the keratin, second one is the collagen, and third one is the elastin. Keratin, it is present in exoskeletal structure, for example hair, nail and horns. Collagen, it is present in the connective tissue, for example bone. And the third one is elastin, which is present in elastic tissue, right? So from this elastic, this word is derived, that is elastin. So it is present in elastic tissues such as tendons and arteries. So these are all examples of the simple proteins. Next we are going to look at the examples of conjugated protein. Now earlier we had talked that conjugated proteins are the simple proteins plus non-protein group. When these two combines, we get a conjugated proteins, right? So simple proteins, we can also call them as a apoprotein. Its non-protein group, we know them as a prosthetic groups, and when these two combine, we get a complete protein. So we can also call them as a holoprotein. Now based on what kind of prosthetic group is present, we can further subclassify it. So this prosthetic group, it can be nucleic acid, it can be carbohydrate, it can be lipids, it can be phosphates, it can be metals, or it can be colored compounds. So let's first look at what will happen if the prosthetic group is nucleic acid. So when nucleic acid is there as a prosthetic group, we call such protein as a nucleoprotein. Now here we have two choices for the nucleic acid. This nucleic acid can be DNA or it can be RNA, right? So if DNA is there, we can call them as a deoxyribonucleoprotein. And where does it found? This deoxyribonucleoprotein, where does it found? It is found where DNA is found. So they are found in nucleoli, mitochondria and chloroplast. Now instead of DNA, if RNA is present, we can call them as a ribonucleoprotein. Now where does this ribonucleoprotein found? Of course, it is found wherever RNA is found. So ribonucleoprotein, it is found in nucleoli and ribosomes, right? Now the second type of prosthetic group which can be present is the carbohydrates. Now in case of carbohydrates, there are two important divisions. The first one is mucopolysaccharides and second one is oligopolysaccharides. If mucopolysaccharides is occurring as a prosthetic group, we can call such conjugated protein as a proteoglycans. And if it is oligosaccharide as a prosthetic group, we can call such protein as a glycoproteins. Now remember, proteoglycans, they are also known as mucoproteins or mucoids. And these glycoproteins, they does not have any other alternative names. Examples of glycoproteins are immunoglobulins, complements, mucins, and many different enzymes, right? Now, if prosthetic group is a lipid, then we can call such conjugated protein as a lipoproteins. Now, prosthetic group can be a phosphate, and whenever phosphate is as a prosthetic group, we call such conjugated protein as a phosphoproteins. Now, here one important problem is there. What is that problem? See, in case of nucleic acid has a phosphate moiety, right? In case of lipids also, if particular lipid is phospholipid, then such lipoprotein also has a phosphate group in their structure. So here one important problem is created. What is that problem? The problem is that in case of lipoproteins, there is a phosphate group. In case of nucleoproteins, there is a phosphate group. So should we call nucleoproteins and lipoprotein as a phosphoprotein? The answer is no. We cannot say. Why? Because one more condition is added. What is that condition? The condition is phosphoric acid or organic phosphate should be present, but not the phosphate containing substance such as nucleic acid and phospholipids, right? So in case of DNA or in case of nucleic acid, there is a phosphate, right? But we cannot call such conjugated protein as a phosphoprotein. We have to call particular protein as a phosphoprotein when phosphate is independently present in particular protein, not as a component of nucleic acid and phospholipids. I hope that this distinction is clear. Now see, this is the apoprotein part of this phosphoprotein, and this phosphate non-protein group is attached with this apoprotein. So when it is attached, it has one characteristic that it binds in this polypeptide backbone, it binds with either serine or threonine. So we can say that serine and threonine residues of apoproteins are involved in binding with phosphate. Now, are there any example? Yes. One example is the casein, which is phosphoprotein which is present in milk, and second example is ovovitelin, which is present in egg yolk, right? Now let's look at the next one, that is metals. What will happen if metals is there as a prosthetic group? So at that time we can call particular conjugated protein as a metalloproteins. Examples are ferritin, where iron is a metal group. Second example is the carbonic anhydrase, where zinc is acting as a prosthetic group. Third example is the ceruloplasmin, where copper is acting as a prosthetic group. Now the last one is the colored compound. Colored compound can also act as a prosthetic group in case of conjugated proteins. So when colored compound occur as a conjugating group or prosthetic group, we can call them as a chromoproteins. Chromo means color, right? For example, we have hemoglobin, cytochromes, catalase. In all these three compounds, heme is a prosthetic group, so they are also known as hemoproteins, right? Then we have certain flavoproteins where riboflavin is a prosthetic group. And the third one is the visual purple. In this visual purple, carotenoid is a prosthetic group, right? So with this, I complete the examples of simple proteins and conjugated proteins. The examples and discussion about the derived proteins, I will cover it in the next video. So I hope all the aspects of the classification of proteins is clear. If you have any query or confusion, please write it down in the comment section below. Thank you.